soft exhale of steam like a long contented sigh across a shallow sheet of water. The colours lie in calm ribbons, pale blue in the pool, then a band of satin green, then a dusky orange brushed with gold. The light is gentle, the sound is low. You can feel yourself breathing in a slow, even rhythm while we wander patiently into the understated life of microbial mats, where metabolism moves like a lullaby and the stories unfold at a very unhurried pace. If this gentle space comforts you, a quiet tap on like and subscribe helps more people find restful science, and the playlist in the description can carry you further while you drift. How sleepy are you on a scale of 1 to 10? Now let yourself sink into the scene. You are at the calm margin of a thermal pool. The stones beneath your imagined feet are smooth with fragile coatings of silica. The water is clear, but you notice that what looks like a velvet carpet clings to the channel floor, following the slope of the outflow like a tender braid. This is the mat, as thin as a folded leaf in some places, thicker and softly rippled in others, made of countless cells woven together with slim strands and gel-like coatings. The steam brings the faint clean scent of minerals, like wet porcelain, touched with a suggestion of earth and a whisper of warmth. There is no hurry here. Even the bubbles that rise from the deeper pool do so with a steady, sleepy cadence. In this unbroken calm, we will keep time with the subtle lives that paint these colours, lives that are content to move slowly, to harvest light with careful hands, to share food across layers, to knit themselves to the rock, to wait and to breathe. Imagine bending close, the way you might lean toward a page in dim light. The colours are not just decoration, they are gradients, signs of temperature and light, signals to anyone who has learned to listen with their eyes. Where the water comes out hottest, the colour may be almost absent, a pale satin sheen where heat-loving bacteria hold fast in a realm too intense for delicate pigments. Just a little farther, the green appears, deep and full, often the hallmark of cyanobacteria, the quiet workers of oxygenic photosynthesis. Farther downstream, in cooler water and gentler light, the mat deepens to browns and oranges, the shades of different pigments and the small particles of minerals that catch in the stickiness. These colours are not painted once and left. They are refreshed breath by breath, day after day, very slowly, a brushstroke made of lives that divide patiently and pass on their tiny harvests. You can picture the life within as a layered neighbourhood. The surface is thin, only a fraction of a millimetre, yet in that small thickness lie gradients of light, oxygen, hydrogen sulfide, and pH. The topmost microlayer is flooded with light in the day, and its residents feast on photons with serene efficiency, turning light into chemical energy, carbon dioxide into sugars, and giving away oxygen as a calm, rising exhalation. Beneath them, another layer receives less light and less oxygen. It feeds differently, savouring the leftovers, using light that filters filters through like late afternoon sunlight through tea. Beneath that, in tiny pockets and channels, oxygen falls away and other chemicals rise, and their other microbes live well without oxygen at all, breathing sulfur instead, or fermenting the day's harvest in the soft dark. All of this is held together by a gentle glue. If you could touch the surface and not disturb it, it would feel slick and a little yielding, like a soft rubber made by life. That texture is the work of exopolysaccharides, long sugar chains exuded by cells, a matrix that traps bubbles, grains of sand, ions drifting in the water, and even warmth itself. Inside that cushion, threads of filaments align to the flow, like hair combed by a stream. Thin channels form and reform as gas moves and the mat breathes. The mat is not solid stone, though it may become a template for stone one day. It is a living blanket, woven of microscopic bodies, coiled like nests, latticed like lace, and always, always slow. In the early morning, before the sun has risen high, the mat feels the day arrive through changes so small that you would barely notice them if you were not watching closely. As the first light folds over the pool, pigments activate with gentle certainty. The cyanobacteria, rich in soft green chlorophyll and protective pigments the color of tea, begin to use the light. Their little factories, the thylakoids, awaken and shuffle electrons calmly from one carrier to another. The sugars made at dawn find their way towards storage granules, kept safe for later, and oxygen begins to rise through the mat. You can hear nothing of this, but you can imagine the quiet effervescence that appears by mid-morning as tiny bubbles form and collect, bright little beads under the surface, like gleaming seeds in a fruit. The oxygen tension grows at the top and pushes downward a little, softening the border of the oxygen-free zones beneath. Deeper in the mat, where the light is dim and colored by the pigments above, anoxygenic phototrophs stir, those that do not split water and do not make oxygen. They dine on light as well, but they feed on different chemicals. Some relish sulfide, turning it into elemental sulfur or sulfate, while they knit their own sugar chains. Others use molecules like hydrogen or organic acids that seep down from the activity above. Their pigments capture different wavelengths, often the light that slips past green harvesters, light marinated in green and leaving more copper-colored hues. They gather it in quiet contentment, as if satisfied to wait for what passes through. This layered feast is not only about light, it is about time. The mat lives with a daily rhythm, a persistently repeated cycle of day and night, and every resident has learned to dance at an appropriate tempo. During the day, photosynthesis reigns near the surface. At evening, as light wanes, oxygen production slows and stops, while oxygen consumption continues deeper in the mat, and soon the surface becomes a haven for fermenters and anaerobes that had waited for the night. Cyanobacteria on top become different in their behavior after dark. They start to ferment the sugars they stored in the gentle light, releasing small molecules like acetate, hydrogen, and ether one group's leftovers become another's supper. The oxygen disappears first from the shallow layers and then withdraws further, like a tide, leaving niches to be filled until the next dawn. Slow metabolism carries a kind of hush. You can feel it with the mind's touch. A single cell here may divide only after several days when the conditions are right, or even weeks. It's life a meditation of resource gathering and careful share. The mat grows, but not in a hurry. It achieves thickness over months and years, its surface worn and renewed, its underbelly compressed and changed. Little filaments move over time, gliding across each other without legs or fins, guided by phototaxis and chemotaxis, moving toward gentle gradients, though you would never see it happen unless you watch a time-lapse that compresses weeks into seconds. Even then, the motion is courteous, layered, a hum rather than a shout. You may imagine a water molecule starting far underground, warmed slowly as it passes through porous rock, dissolving minerals, learning the taste of silica, sodium, and carbonate along the way. Its path takes time, and the heat is gradual, a tender cradle rather than a blast furnace. When it emerges at the vent, it is clear and shimmering. It carries a little gift basket of dissolved minerals, which will be offered to the mat as the water cools. Silica may precipitate in tiny spheres that gleam with a glassy sheen under a microscope. Here and there these spheres settle between the filaments, like soft sand, adding gentle weight, lending the mat a quiet memory of stone. In carbonate-rich systems, the precipitate is different, a chalky microcrystalline travertine that settles in feathers along the edges of bubbles and filaments, turning organic textures into early fossils, as if copying the mat's handwriting into a ledger of rock. If you could shrink down and dwell for an afternoon at the top of the mat, you would feel light passing through your own body. Even your shape would matter, the way your pigments are arranged, the microarchitecture of your thylakoids, the angle of thin lamellae. The light would be warm but filtered, changed softly by the pigments above and below, as if lying under leaves near a pond. You would sip carbon dioxide that diffuses in from the water above and tap into a small bank of inorganic carbon, the diffuse pool that changes with the chemistry of the spring. You would protect yourself with pigment shields like cytonemin, a sunscreen made by some cyanobacteria that sits in the sheaths and gently dampens the harsher parts of sunlight. You would ramp production up as clouds move and down as they pass, but nothing would be abrupt. The mat is a place without sudden jumps, its inertia smoothing the sharpness of the outside world. When midday comes, oxygen bubbles gather and line up along channels in the mat. They push gently, opening slits, stretching thin roofs of gel and filaments, then slipping out to wander toward the surface of the water. Each tiny channel is a pathway for breath. If you imagined placing an invisible needle into the mat, a very fine probe that measures oxygen concentration, you would see a curve that climbs at the top, then falls sharply just a fraction of a millimeter down, then drifts toward zero deeper in the serene interior. During the brightest hours, this oxygen penetrates more deeply, but it can never go downstream, where temperatures fall into the range that suits heat-loving cyanobacteria, the mat becomes rich with green, often deep emerald at first, then softening. Further still, as the water cools to a temperature more welcoming to broader communities, the browns and oranges arrive, coloured by accessory pigments like carotenoids and by the fine mineral grains that settle out as the water loses its heat. These colours gently signal the temperature, a thermometer that you can read with your eyes from a distance, and the transitions are gradual, never abrupt, matching the nature of the flow itself. You may think of Yellowstone and the famous outflow channels that look like long ribbons of silk laid over stone. Names like Octopus Spring and Mushroom Spring carry their own poetry. There, populations of cyanobacteria, adapted to different temperatures, line themselves up, each in its preferred band, a tidy arrangement like shelves in a quiet library. Within the bands, different lineages coexist, some preferring more light, some less, some more tolerant of the oxygen that accumulates in midday, some relying more on nighttime fermentation. The bands appear still to the eye, but they carry cycles within cycles, daily rhythms woven into seasonal rhythms, woven into years in which the hydrology may change, the mineral composition may drift, the vent may pulse, a little warmer, then cooler, a slow heartbeat that the mat feels in its bones. If you kneel at the margin and look carefully, you might see that the surface has a nap, a texture like the velour of a worn sofa, with short filaments that lift and lie down with the flow. Each filament is a chain of cells, sharing wall to wall, the whole chain able to glide by secreting and rearranging polymers along its surface. They align themselves to the direction of water, become streamlined, and in doing so create an organized porosity that lets chemicals diffuse more predictably and light transmit in a feathery way. In places the mat forms small domes, little mounds where gas has lifted the surface, then escaped, leaving a hummock with a soft crest. In other places, the mat drapes over a pebble and curves like cloth, making a calm, rounded shape. These micro-topographies create tiny microclimates, a shadow here, a brighter pocket there, a place where oxygen gathers a bit more, a fold where anoxygenic phototroughs find a small sanctuary. The chemistry that flows through the mat is a slow ballet. Carbon moves in as inorganic carbon, mostly bicarbonate in these alkaline waters, and is fixed into organic forms by the day's light work, then handed off as the day wanes. Nitrogen may drift in as nitrate from the spring water or the air, and some bacteria hold the skill to fix nitrogen gas directly, making ammonium that feeds the entire neighborhood, especially at night when oxygen recedes, and these delicate enzymes, so sensitive to oxygen, can work in peace. Phosphorus arrives attached to tiny particles or dissolved as phosphate, and can bind to the exopolysaccharides, entering a soft exchange bank where it can be accessed as needed. Sulfur arrives as sulfide in springs that pass through sulfide-bearing rocks, and that sulfide becomes a quiet linchpin for communities that use You can follow a single molecule of hydrogen sulfide in your mind. It drifts upward from the water beneath, reaching a layer filled with phototroughs that use it as an electron donor. It is captured and oxidized gently, leaving behind elemental sulfur that clings in tiny granules for a while, perhaps to be oxidized further to sulfate by neighbors, perhaps to dissolve again. The act of eating the sulfide makes the layer safer for oxygenic photosynthesizers above, because sulfide in too high a concentration would harm their delicate machinery. In turn, the oxygen made above supports communities a little deeper that can use it to oxidize leftover compounds. Mutual comfort emerges from this layered dining. No one speaks, but everyone signals with chemistry, and the mat hums with cooperation. A mat like this can teach patience. You can imagine yourself sitting at the boardwalk, the wood warm beneath you, your shoulders eased, and your eyes half closed, feeling the consistent low murmur of water. You think of how many cycles have passed to build even a few millimeters of this fabric. You think of how, under certain flow regimes, silica has begun to fix to the outline of filaments, turning their shapes into mineral shadows. Over time, sheets of sinter can accumulate. The structures have names that are soft in the mouth: palisade textures, shrub textures, spicules, streamers. Some are made in different conditions, especially in very hot, fast outflows closer to the vents, where mats are sparse or shaped into long filaments called streamers that flutter gently. In cooler terraces, travertine or calcite may build shelves that look like bath steps for a miniature world, and the mats drape over them like living cloth. In other springs, perhaps more acidic, life still finds a way, but in different costumes. Acidic waters often favor iron-oxidizing bacteria that form streamers and flocks, their colors closer to rust and cream than to green and orange. The chemistry of acidity changes what pigments survive and what enzymes keep shape. Yet even there, the principle is similar. A gradient, a slow, soft parade from the vent outward, from heat and chemical intensity toward gentleness, with different lives stepping in at each point, content with their niche, nothing hurrying, nothing starved for speed. A day in the mat has many quiet chapters. In the morning, the light arrives creamy and welcoming, and cells activate their photosystems, balancing the two main streams of electron flow, cyclic and non-cyclic, tuning as calmly as a musician tuning strings. They protect themselves from too much light with harmless heat dissipation and with pigments that act as soft shields. Midday brings the highest oxygen levels, a rise that may push the surface into supersaturation, giving life to gas bubbles that look like strings of pearls. The vibrations of the flow may wick these pearls along little channels, and as they pass, they may leave rings of mineral behind. Afternoon settles in with a long, calm decline in light, and you can imagine the whole mat shifting its metabolism like a community that closes its shutters for the night, preparing for quiet. The oxygen is consumed faster than it is made, and pockets of darkness come alive with fermentation and anaerobic respiration. Sugar granules accumulate lies wait hungry. Before dawn, the mat is at its most breathless state, oxygen gone, the gradient flat with night calm. Then the first photons of morning arrive, and quietly, predictably, everything resets. The idea of slow stories suits these mats well. Over weeks and months, the surface can subtly shift in color as different populations adjust to changes in flow or seasonal sunlight. In summer, when days are long, the phototrophic layers thicken and darken, and in winter, under snow or shorter days, they thin and pale, crisper but still present, warmed by the spring. If a heavy rain changes the flow, it can peel away part of the mat, but even that disturbance has a gentle edge in this narration. The mat remembers how to grow back. Cells, gliding from edges, begin to knit a new layer over exposed stone. Spores or resistant cells that endured the flush awaken, and bare patches become veiled again. In months, a smooth surface returns. In years, thickness grows. In decades, mineral castings record the presence of those earlier communities. Nothing is wasted. Nothing is done with haste. As you let your thoughts drift, you can picture a scientist holding a small transparent core, a bit of mat captured in a slender tube. In the lab, this core is kept warm, and needles as fine as hair measure its interior chemistry. An oxygen microelectrode draws a smooth curve on a screen, tracing the breadth of the mat as clouds pass over a lamp that simulates sunlight. Another probe measures pH, revealing small dips where acids appear during fermentation. Yet another captures sulfide profiles below. Together, these curves describe a living architecture more clearly than any photograph. They show that life has arranged itself in steps, like a gentle staircase carved in the thin dimension of millimeters, each step a place where a different process is most at home. Observing this, the scientist does not need drama. The data feels like the trace of a sleeping creature's breathing, regular and deeply reassuring. When you imagine the sunlight across the mat, you might also imagine the way pigments absorb different colors. Chlorophyll takes in reds and blues, letting green appear to your eyes. Carotenoids gather light too, but they also quench excited states that could hurt, functioning as a kind of yawn that releases the extra energy as warmth without harm. Some mats carry cytonemin in their sheaths, a pigment that is resistant and enduring, made when ultraviolet light is intense. Cytonemin turns the mat surface a more brownish tone, a patient umbrella for cells underneath. Other pigment families add gentle hues like phycocyanin and phycoerythrin in some cyanobacteria, phycobiliproteins that give blue-green or red tints, shifting the palette very slightly, just enough to notice if you look carefully. On a partly cloudy day, these pigments help the mat adapt to changes in light, a soft buffer, like an extra blanket placed on or taken off with slow hands. There is a measured resilience in the mat's everyday life. A little grit lands from the wind and becomes part of the fabric. A small change in water chemistry introduces more silica for a while, and the mat grows slightly stiffer as gel. A scalding pulse of hotter water arrives one afternoon when a deeper channel opens temporarily and sensitive surface layers thin, but deeper layers endure, and over weeks the surface returns, repopulated from edges and below. In another place, a cooler pulse flows, inviting new organisms that prefer that gentler temperature, and the band of green shifts a few centimeters downstream. This slow movement is not migration in the dramatic sense. It is adjustment through growth and attrition, a patient eddy in a living river of time. Consider the role of the viruses that drift through, particles as simple as a small box with a spool of genetic thread. They enter cells and replicate, then leave again, sometimes with pieces of genes that move between lineages. This gene exchange is another whisper of change, a way the mat keeps a library of solutions to small problems, with such gentle pressures, genes that help in this very specific environment spread, not like a fire, but like a quilt slowly extended one patch at a time. The mat's community is not fixed, but it also does not veer wildly. It is a drift net that catches what works and lets go of what does not. Human curiosity finds its own slow harmony here. Decades ago, a bacterium isolated from a hot spring gave us a useful enzyme, a DNA polymerase that does not unfold at high temperatures, allowing a form of DNA copying to proceed through heat cycles without losing its helper. That bit of molecular patience carries a quiet connection to the mats we are visiting in our imagination. The same stability that lets life continue at 60 or 70 degrees Celsius helps hold together a reaction that people now use in labs and clinics. It is an example of how slow, persistent life in a warm spring offers steady gifts to the wider world. Behind the scenes of every surface, you can see lies a history older than mountains. Mats like these, or something like them, probably carpeted shallow environments on the early earth. Their layered structures, captured in ancient rocks called stromatolites, record cycles of growth and mineral deposition, laminated a bit like pages in a low, rounded book. In some places today, mats in marine lagoons still make stromatolites as they trap and bind sediment and encourage minerals to settle. The hot spring counterparts here do similar work with silica and carbonate, writing their presence in microtextures and small domes and ridges. Through them, the pace of the past becomes more understandable. Long periods of quiet accumulation, interrupted still. Around a bend in the channel, the colour changes again. Here, the flow is slightly faster, and the matte surface is smoother, the texture tighter. The filaments align more perfectly, a sign that shear stress is different here. The bubbles cannot linger and are whisked away, leaving the surface matte rather than beaded. Downstream, where the flow broadens and slows, bubbles gather, and small cabbage-like rosettes of silica begin to form around the edges where oxygen-rich microenvironments favour precipitation. Over weeks, these rosettes build a thin crust that is flecked with life. The mat in these sections is thinner, but can cling to the underside of the crust, building a little sheltered space that persists even when the flow registers small changes. You can think of these spaces as tiny verandas built by cooperative effort, lived in quietly. If you scoop a small cup of the water in your mind and look at it closely, its clarity hides complexity. Dissolved silica content might be hundreds of parts per million, held in a metastable, quiet state at the high temperature, ready to settle as the water cools. Sodium, potassium, chloride, and bicarbonate are there too, balancing charge, making the water taste salty if you dared to sip. Trace metals in minuscule amounts drift through, and the mat, being sticky and generous in surface area, captures some and lets others pass. The water is a conveyor belt of gentle offerings. The mat takes what it needs and makes do with the rest. Sometimes late in the day, the light angles in and reveals a gloss on the matte surface, a sheen where exopolymers reflect with a soft shine. This natural gel protects against desiccation when a small change in flow leaves a patch exposed for a while. It binds particles, it holds cells together against the current, it reduces friction and allows gliding. The gel has structure at the nanoscale, with binding sites and crosslinks, but to a casual glance it is simply smooth and reassuring. In the lab, you could dissolve it carefully and find long sugar chains, interesting linkages, carboxyl groups that can bind metals. In the field, you can only sense its function in the way the mat feels whole, neither flaking nor flapping in the flow. Further downstream, you notice patches where the mat is not continuous. Small grazers live there, creatures like tiny snails, caddisfly larvae, rotifers, maybe even tardigrades, all in sizes too small to see at this scale of viewing. They move at their own slow pace, grazing and gently cropping the surface, changing the texture in small patterned ways. In many springs, temperatures are too high for these animals, so their influence is limited to cooler fringes. Where they do appear, they create small mosaics in the mat, an extra texture of use and regrowth. And even here, the tone remains soft. Grazing is not a harsh intrusion, but a quiet part of the larger fabric, balanced by the mat's ability to grow afresh after being trimmed. Imagine rain pattering on the boardwalk roof, the sound muffled in his roughness to suit the average flow. The idea of homeostasis, of keeping internal conditions within a gentle band, plays out across the mat, not with sharp controls, but with distributed responsiveness, a mat-wide consensus about comfort. In the late afternoon, you think about how information moves here. There are no nerves, no wires, but signal molecules pass from cell to cell. Small peptides may tell neighbors to make more or less matrix. Quorum sensing signals rise when density increases, prompting a shift in gene expression, a change in how often a cell divides, a nudge toward forming a thicker surface or a thinner one. Phage infections wax and wane, their lytic episodes perhaps trimming back a patch of overgrowth and making room for diversity. Gene exchange through plasmids and transducing phages offers a slow roll of adaptation, not frantic, but present. A mat grows, a mat learns, a mat remembers through lineage and through shared chemical streams. You may recall the graceful names of microbes that call this place home. Synechococcus and other slender cyanobacteria glide near the surface. Chloroflexus, green non-sulfur bacteria, live just below, using near-infrared light and oxidizing various donors, while sometimes also indulging in photoheterotrophy, sipping organic compounds provided by neighbors. Rosiflexus, oscillatoria-like forms, and many unnamed lineages contribute as well. Deeper in, sulfate reducers make hydrogen sulfide, cycling sulfur along a course that arcs back up through oxidizers. Methanogens capture hydrogen and carbon dioxide in the deepest anoxic corners and make methane in modest quantities, adding another gentle layer to the breath of gases that pass through the mat. Nitrifiers may appear where oxygen is present. Denitrifiers pick up the threads where oxygen falls away. It is a woven suite of metabolisms that interlock like fingers, and the handshake is steady. At night, the stars above the spring wood, in a cloudless sky, mirror themselves faintly in the water. The steam might veil them, but not entirely. The mat below, now in its anaerobic phase, continues to share quietly. Fermentations produce small organic acids. Hydrogen diffuses in smooth gradients, satisfying hydrogenotrophs below. The stored sugars dwindle and are replaced next day. The daily meter keeps time, and below even this beat lies a deeper metronome in the cells of cyanobacteria, a molecular circadian clock made from proteins that assemble and disassemble with a roughly 24-hour rhythm. These timing mechanisms help schedule metabolism and cell division so that oxygen-sensitive processes happen in the soothing blanket of night, and oxygen-producing processes bloom under the day's safe watch. It is attuned so well learned that even when the day length changes, the cells keep a nearly steady rhythm, drifting slowly until external light nudges them back into phase. You can see, in your mind's slow scan, the path of oxygen bubbles as they wiggle loose from surface films and rise. You can see the way a slight change in flow causes fine sand to settle in the mat's shallow dimples, or to be lifted and carried further. You can see the shimmering effect, called the heat haze, rising from the water soften your gaze and let the edges blur a little, so your mind can rest on the big shapes and the slow movements, rather than on details that snag attention. When people measure the growth of a mat, they sometimes place little markers and return after weeks to see how far the mat has advanced. The figures that result are modest, millimeters over months, less in winter, more in summer, depending on the spring. In a culture dish, under more generous and controlled conditions, growth can be quicker, but here in the field, the mat abides by its own quiet economy. It is this slow economy that keeps the structure so cleanly layered, that gives us the soft, stepwise chemical profiles we can measure, and that lets mineral textures build predictably. Speed would make a jumble. Slowness makes a pattern. The understory of this narrative involves the rocks below, which in many hot springs are rich with silica, formed by volcanic activity that has left its heat in the ground. Water percolates through these rocks and dissolves silica as it goes. When it emerges and cools, the solubility of silica drops and microcrystalline opal precipitates. The presence of the mat provides many nucleation points, tiny invitations for the silica to settle. The exopolymers slow diffusion and give space for delicate silencers of turbulence. In that shelter, minute structures form, spheres, collars, fans, according to modes of precipitation and flow. These structures may preserve the shape of filaments and sheaths. Over time, whole tracts of mat are replaced by opaline silica, leaving crisp replicas that you could see in thin section, a frozen record of life's gentle carpet. If you imagine returning to this place in 100 years, you could see a new layer of terrace built up ever so slightly, and if you looked within the stone, your eyes could trace fossilized filaments that match the ones living now. For a moment, let your thoughts move to a distant analog where similar processes occur at different scales. In shallow marine lagoons with high evaporation, microbial mats trap sediment and encourage carbonate to precipitate, building stromatolites that grow very slowly. The idea of layering across time and the idea of cooperation across metabolisms are shared between these marine mats and the hot spring mats. Together, they tell a story of early life strategies. Build community, shape your environment in small increments, and leave a record in stone when the opportunity arises. The record is not in loud parentheses, but in gentle underlining, laminations among which you could run a fingertip and feel each step of an ancient staircase. Still awake? Comment the secret word, nebula. Bringing your attention back to the hot springs margin, you might gaze into a thicket of long waving streamers near a hot event, where mats as we described are not flat carpets, but bands of filaments attached at one end, fluttering. These streamline below a thin line, where temperatures suit only a few specialized thermophiles. The streamers are often built by bacteria that oxidize reduced iron or sulfide, and they carry their own microgradients. Even at these high temperatures, not every piece of the flow is the same. The edges cool just enough to let different lives take hold. Flow is not uniform either. Very small eddies and shelter zones along the sinter surfaces create microblind places where quiet pools form and different communities can rest. These micro-eddies become petite laboratories, replicates of the larger world in miniature, complete with daily cycles and chemical handshakes. In gentle words, we can scoot beneath the surface with our thoughts and visit the middle layers in more detail. Chloroflexus, a common resident in some hot spring mats, uses a type of photosynthesis that is different in a subtle, satisfying way. It does not make oxygen and often uses organic compounds or hydrogen as donors. Its pigments absorb in the near-infrared, a range of light that is plentiful just below the green layer, filtered like sunset light filtered through leaves. Chloroflexus can also switch to different styles of metabolism depending on the day's offerings, a flexible eater in a neighborhood that prizes versatility. Rosiflexus and other relatives have similar talents, each with its own quiet fingerprint. The surface layers, in turn, give them a landscape of light that changes through the day in small, predictable ways. Morning has one flavor, noon another, evening a third. These small flavors guide shifts in gene expression, not with a sudden click, but with a slow slide. You can listen, in a figurative way, to isotopes speaking softly in the mat. Carbon isotopes carry a fingerprint of the way carbon is fixed. Nitrogen isotopes whisper about the balance between fixation and consumption. Sulfur isotopes tell of sulfide oxidation and sulfate reduction. By measuring these variations, scientists have learned to trace the paths of elements through the mat. This kind of science is like eavesdropping on a distant conversation over a thick wall. The words are blurred, but the rhythm and tone can be discerned, and with careful analysis, the topic becomes clear. Again, there is no drama, only the steady accumulation of understanding, like silt settling from a quiet river. Everything about the mat suggests that smallness and slowness can be comfortable. The mat does not chase opportunities, it waits where the flow reliably brings them. It does not build towers, it weaves a quilt. It does not demand attention, it rewards gentle observation. Sitting with it in thought feels like sitting under a warm blanket with a book you do not have to finish. You turn a page when you like, you pause to sip water. The story is patient. Time in this narration continues to stretch out softly. Afternoon becomes evening. The sun leans toward the horizon and the colors warm. On the boardwalk, a dragonfly perches, and a breeze moves a curl of steam like a veil. You can think about how in winter, the cold air makes the steam thicker and more dramatic, while the mats below continue their work untroubled by air temperatures that would be harsh elsewhere. The water keeps them cozy, a constant bath that insulates and comforts. Snow may dust the edges, turning the terraces into soft shapes, but the mat's daily cycles continue with only minor adjustments. The pace is the same song in a different key. In such a setting, the idea of a disturbance takes a muted form. A branch falls across the channel after a windy afternoon and slightly damps the flow, creating a small pool behind it. The mat thickens a little there over weeks and thins a little downstream, adjusting like a gentle breath. A visitor accidentally drips a few drops drops of sunscreen in the water, and the mat surface beads it and lets it pass in broken little patches, and then everything is as it was. A splash of sediment from a rainy day coats a section, and the surface carefully lifts itself back into the light, the way a plant might push through a thin layer of leaves, only slower, only in millimeters. The mat teaches that most changes are not collisions, but caresses, and that time smooths rough edges. You may enjoy the thought of the mat's sensory world, where light is not a blast, but a range of tones. Some cells know the direction of light and slowly orient themselves to it. This capacity is called phototaxis, and it is executed by redistributing cellular machinery and by secreting or absorbing matrix to create a slight movement. The movement is measured in micrometers per minute or slower, making a scale of time that feels like a lullaby compared to the tempo of our daily life. Chemical gradients are felt through receptors that bind the molecules as they drift along. The mat has many little ears, and they listen patiently. The vocabulary of energy here is straightforward yet elegant. Photons are absorbed, electrons are excited, proton gradients are established across membranes. ATP is made, the currency of the cell. The mitochondria do not appear here in the same way they do in larger organisms. Instead, membranes inside bacterial cells handle the job. The proton motive force hums like a silent motor, powering the slow synthesis of sugar and the careful folding of proteins. Even the names of the proteins can sound restful when said softly. Ribisco, the enzyme that takes carbon dioxide and binds it in the Calvin cycle, has a bucolic ring, and although it is famously not the most efficient enzyme, here in the mat it is sufficient and dependable. The inefficiencies are part of the charm. Excess is not necessary. The mat persists with good enough solutions layered together. If we tune our senses to the geology again, an interesting detail appears. Silica sinter tends to grow outward as thin films that crust over surfaces and trap tiny bubbles, producing a lace of round holes or small bridges called fenestrae. Under the mat, these spaces keep microhabitats open. When a bubble forms and resorbs, a small dome of sinter is left behind. Over long periods, these domes accumulate into patterns that seem almost botanical, like dried seed pods. Travelers who visit ancient silica terraces find these patterns in stone, with no living mat. The record says that a mat once lived here, with patience and rhythm, and then conditions changed, and the mat moved on or the spring's plumbing shifted. The details remain preserved, quietly elegant. Elsewhere, carbonate terraces build themselves step by small step, encouraged by degassing of carbon dioxide as water cascades in thin sheets. The geometry of these steps affects flow, and with it, affects where mats take hold. On the upcurrent edge of each rim, water is narrow and swift, discouraging thick mats and favoring thin films. In the lee, water spreads and slows, encouraging thicker mats with more visible textures. In such a terrace system, tiny forests of carbonate needles can grow, where mats hold the water still enough for precipitation, and the mats grow around and through these crystals, making a You may consider for a moment the global tapestry of hot springs. They exist where tectonic plates pull apart, where volcanoes sit, where crust is thin. Iceland, New Zealand, Japan, Kamchatka, the western United States, parts of China, even remote places in the Andes and the Ethiopian Rift, all host warm waters where mats can draw their slow lives. Each region has its particular chemistry and its local palate. In some, arsenic is more abundant, and mats grow that can handle this with smart pumps and detoxifying enzymes. In others, boron presents, or different proportions of sodium and potassium. These differences colour the lives of the mats in subtle ways, shaping which lineages thrive and what pigments they prefer. Yet, standing at the edge of any of them, you would recognize the same essential calm, the same gentle flow, the same preference for gradual change. A soft memory from the history of science whispers through this place. Early observations of microbial mats in hot springs led to the recognition that some bacteria could live at temperatures that seemed too warm for life, as people then imagined it. The word thermophile entered the public imagination. From there, an ever-widening appreciation of extremophiles followed, organisms thriving in salt, acid, or pressure that would unsettle most life. In that story, hot spring mats have a quiet starring role, not as spectacular performers, but as the steady backdrop that made discovery possible. It is nice to think that patience itself drew back a curtain and revealed a broader stage. Tucked into the mat are pockets of gas that come and go. While oxygen dominates bubbles during midday, other gases pass too, like nitrogen and carbon dioxide. Methane may catch in small amounts in deeper parts, a minute addition to the mix. When you peer at a bubble under a microscope, you see the way it bulges against the mesh of filaments and polymer, stretching but held in place, a patient bubble that waits for enough pressure to slip free. Each bubble is a punctuation mark in a sentence, without haste. When it leaves, a tiny crater is left behind briefly, and then smoothed over by gliding filaments and the slow secretion of exopolymers, like a pillow fluffing itself back into shape. Night returns in our mental picture, and with it an invitation to rest. The mat, dark at the surface now, still devours energy in the form of chemical reactions in the lower layers. This consumption and the breeze across the water surface can cool the mat slightly. The spring's water warms it again from below, creating a small, cozy gradient, warmer at the bottom than at the top, a thermal blanket effect. In this peaceful microclimate, processes that disliked oxygen get their turn. Sulfate reducers remind us that the sulfur cycle is ancient and comfortable here. Iron reducers and methanogens do their part in the deeper microlabyrinth. When day returns, oxygen creeps down again and calls a gentle pause to their strongest activity. But the continuity remains. The mat never truly sleeps. It drifts through modes, like a person in different phases of rest, all restorative, all part of the whole. When you lift your eyes in your imagination and look across the pool, you see the grander architecture structure of the spring. Pools and channels link like small lakes connected by streams. The mats write their colours along the map. In places where the channels split, the mats choose both paths and then diverge into new patterns as the chemistry and flow change. The system is a quiet experiment, repeated thousands of times by nature, exploring how life arranges itself in response to subtle shifts. For those who love to learn, it is as satisfying as watching a garden change across a season, an invitation to come back and look again without pressure. If one were to place sensors across a mat and record for days, the curves would reveal the circadian rhythm of this community. At midday, the oxygen at the surface might rise to 3 times air saturation, then decline toward evening. At night, pH in the deeper layers would gently drop as acids accumulate, then rise with the new day. Tiny dips and bumps in these curves would align with clouds passing, with the passing shadow of a tree, with subtle variations in flow. The mat is a sensitive instrument, and yet it does not tip over with slight changes. It simply sways like grass in slow wind. There is a comfort in knowing that life can take on this shape. Not everything needs speed. Some systems are built on patience and reciprocity, on carefully tuned gradients, on a willingness to wait. In our world, where speed often receives praise, it is soothing to linger in a place where the virtues of slow metabolism and slow stories bring stability and beauty. It encourages a gentle rebalancing of our own tempo. You can let your breathing match the mat cycle in a symbolic way, in on the imagined day, out on the imagined night, on and on quietly with no need to measure. Moving once more into the details, you may think about proteins in thermophiles that are stabilized by stronger internal contacts, more salt bridges, more compact folds, sometimes anchored by chaperones that help keep proteins in good shape at thermal conditions that would unravel ordinary proteins. Membranes here are built with lipids that pack tighter, sometimes with ether linkages in archaea, making the membrane more resistant to heat. DNA is supported by proteins that prevent it from fraying. These small molecular choices create a physiology that fits the spring's environment. The choices are not flashy, they are simply appropriate, a pleasant word for the right tool for the right job. Under gentle flow, the mat can trap minute bits of organic debris, a stray pollen grain, a sliver of algal cell from a cooler pool upstream. These inclusions are tiny feasts for heterotrophs in the mat, those that feed on organic carbon rather than making it. Their work complements that of phototrophs and chemolithotrophs, completing the loops of carbon flow so that little escapes unused. In this closed local cycle, waste is rare. What one cell discards is another cell's dinner. The mat's economy is circular before the word became fashionable, quietly efficient, almost austere. In some springs, arsenic and antimony are present in trace amounts, and microbes there employ pumps to export these elements when they present as toxic ions, or they reduce them to forms that precipitate harmlessly. These adaptations situations are small acts of problem solving. In others, ultraviolet light is strong at high altitude, and mats there may make more cytonemin, weaving a thicker shade. At lower altitude, where UV is gentler, the shade can be thinner. All of these flexibilities maintain comfort in the face of minor differences, and so keep the soft, predictable rhythm intact. You may picture a morning when fog hangs low, and the world seems wrapped. The pool's edge is a suggestion, and beyond, the pines are soft shapes. In that softness, the mat below remains precise. It senses the light even through fog, a little slower to rise in oxygen perhaps, and a little earlier to sink back into night's mode. Birds call at a distance. Footsteps on the boardwalk arrive in pairs, then stop for a while, then move on. The spring cares little, going about its endless hush. If you ever have the chance to stand at such a spring, you may find that the place lends itself to silence, as if speaking would ripple the surface in a way that is not needed. Among the organisms at the top of the mat, gaseous vesicles may form, small protein-based bladders that adjust buoyancy in some cyanobacteria. In a mat where cells are attached, buoyancy affects position at a very fine scale, allowing cells to adjust their vertical whereabouts by microns. It is another tool to keep each cell near its preferred microenvironment. Those that do not make vesicles may change their carbohydrate content, becoming denser or lighter as they store or consume sugars, shifting position through gliding and gentle pressure. The effect is akin to a very slow tide in the mat's vertical dimension, different niche zones rising and falling slightly through the day. The mat's uppermost layer is sometimes called the photic zone, only a few tenths of a millimeter thick. Beneath it, the chemocline forms, where oxygen fades and sulfide rises, a soft border that moves with the day. Below that lies the anaerobic core. These terms have the ring of big waters, but here they apply to thin worlds. The idea that big ecological gradients can be miniaturized and still hold their character is a thought that comforts, reminding us that scale does not diminish meaning. A forest canopy and a microbial mat share patterns. Light at the top, shade and different guilds below. Nutrient cycles that depend on handoffs and decomposers and mutualists. In periods when a mat is left undisturbed for long enough, its surface acquires a lamination that is visible in cross-section. Thin laminations, sometimes daily, charm. Recall that the mat is not always a flat plane. It grows over stones and into microcavities. It forms bridges. It hollows pockets. It makes a topography that diffuses chemicals in characteristic ways. The shape amplifies the diversity of microhabitats. On the lee side of a pebble, oxygen may linger a little longer into evening, making a niche for organisms that like a longer day. On the upstream face, the rush removes bubbles faster, making it more a place where sheer hardy forms take hold. Across a bump, light angles and shadows form. On the crest, light is bright. In the trough, light is muted and warm. In each tiny place, life sets an appropriate table, never rushed, always ready. It is soothing to think that such mats, which look from an adult height like colourful carpets, are whole worlds when seen at the scale of their inhabitants. To a cell, a micron is a long stroll. To a filament, a millimetre is a neighbourhood. The mat spans centimetres to metres, offering variety within that scale that is as complex as the variety of landscapes on a mountain range. Each microliter of water bathing the mat carries more elements of life than a room full of furniture, yet the overall impression is gentle, not crowded. It is the calm that comes from everything having found its place and cadence. Let us give a moment to the neighbours we do not usually see. Fungi can sometimes be part of the mat, filamentous and quiet, contributing to mineral binding or to decomposition in deeper layers. Protists graze lightly. Small colonies of viruses inhabit the matrix, poised between activity and dormancy. None of these press for attention in the image on a postcard. They work in the quiet, doing small tasks, part of the woven economy. When the mat thickens, the deeper layers may compact and slowly turn over their contents to the steady march of decay, freeing nutrients that can then be reached when the surface thins or when channels open. The mat is in this way its own recycler. In contemplating slow metabolism, we are invited to value efficiency per unit time differently. A quick burst of activity can be wasteful in a place where resources trickle. Here, the trickle is the rule. Energy arrives as sunlight, steady in the day, absent at night. Chemicals arrive in small sips. The mat's inhabitants spread work out across hours, even across days, balancing production and oxygen did not rush in. It seeped and spread over long arcs of time. The legacy of that era remains in our atmosphere, in iron formations that were laid down when oxygen changed the world, and perhaps in lineages that still bear the memory of strict anaerobiosis. Hot spring mats are not direct copies of those ancient communities, but they echo them, and the echo is comforting, like a repeated line in a lullaby. If your mind wants one more small detail before rest, let it settle on the way cells maintain themselves at high temperature. Their proteins are often richer in certain amino acids that promote tighter packing. Their DNA may be supported by stabilizing solutes, small molecules that favor folded states. Their membranes have fewer double bonds, making them less fluid and more resistant to heat. Chaperone proteins act like careful guardians, catching misfolded proteins and giving them a chance to refold. All of this is like tucking in corners of a blanket. It keeps the whole tidy and reassuring. Returning to the mat surface, day by day, the same rhythm continues. A planar wave of light sweeps over at dawn. The phototrophes answer with a whisper of oxygen. The middle layer answers with a gentle shift in its own metabolism. The deep layer listens to the retreat of oxygen and answers in the night. The small trades continue. The community remains. When flow changes, the mat leans into it. When temperature drifts, the mat shifts its bands. When minerals drift and settle, the mat incorporates them in its weave. Such gentleness can be inspiring in its own quiet way, suggesting that we too can find comfort in adjusting slowly to what arrives. Imagine leaving the boardwalk and drifting in your thoughts to a warm room at home. The images of green ribbons and orange bands and soft bubbles drift with you. Your breathing has found a steady cadence, and your eyes feel heavy in a pleasant way. If you like, you can let the mat fade to a cool abstract, a tapestry without labels, simply texture and warmth and light. You can let little details rise and sink at their own tempo, and you can allow your mind to be as unhurried as warm water. Before we close, allow the last notes of this gentle study to settle in. The understated life of microbial mats in hot springs is, at heart, a lesson in generosity, a sharing of energy across layers, a sharing of space across gradients, a sharing of time across cycles. It is also a lesson in the beauty of subtlety, where small differences matter and where stability is built from many small agreements. These communities are easily overlooked at a distance, but up close they are as intricate as any forest, as serene as any pond, and as instructive as any book. They ask very little of us beyond patience, and in return they offer the soothing sense that the world runs on many speeds, and that one of those speeds, a very comfortable one, is slow. As you ease back into your present space, know that you are safe and comfortable, and that rest is welcome. If this soft journey helped your eyelids grow heavy, a like